behalf of the US National Cancer Institute Center for Global Health, we're really delighted to have Dr. Harold Varmus back here in Delhi. Certainly a brilliant scientist, and many of us were lucky to have him as our leader, and certainly one of my own personal scientific heroes. So I'm going to leave his formal introduction to Dr. Chandrima Shah and just quickly thank all of the people who really helped pull this together. Dr. Rath and Dr. Ritu from AIMS, Dr. Vidnukla from the US Embassy, Dr. Shahid Jamil from the Wellcome Trust India Alliance, and of course, Dr. Chandrima Shah. As I mentioned to her, when I ask NII to partner with us on something, I know that it will be executed perfectly, and in fact, this is no exception. So with those few words, Chandrima, please.
Professor Varmus, Professor Talwar, distinguished guests, friends, and colleagues. It is with great pleasure that I welcome you to the National Institute of Immunology. Today's lecture has generated a lot of interest, and particularly among young scientists, budding young scientists of our country. So a special welcome to them. Dr. Varmus really does not need an introduction, but for the benefit of the undergraduate students who have come to attend this lecture, I will briefly introduce him. It is interesting that Dr. Varmus received a bachelor's degree in English literature from the Amherst College and a master's in English from Harvard University. Later, he received his medical degree from the Columbia University College of Physicians and Surgeons, and he did spend some time in India after that. In 1989, Dr. Varmus was awarded the Nobel Prize in Medicine, in Physiology and Medicine, for discovery of cellular origin of retroviral genes. This discovery led to the isolation of many cellular genes that normally control growth and development and are frequently mutated in human cancers. Dr. Varmus is also renowned for his studies on the replication cycles of retroviruses, hepatitis B viruses, and the function of genes implicated in cancer and the development of mouse models for human cancer. He was named by President Bill Clinton to serve as Director of the National Institute of Health, a position he held till 1999. After his tenure at NIH, Dr. Varmus joined Memorial Sloan Kettering Center as President and CEO and served till 2010. Subsequent to this, President Barack Obama appointed him as the Director of the National Cancer Institute. He is a member of the National Academy of Sciences and the Institute of Medicine, and is involved with several initiatives to promote science and health in developing countries. He was co-chair of President Barack Obama's Council of Advisors on Science and Technology and served on the World Health Organization's Commission on Macroeconomics and Health. Dr. Varmus was co-founder and chairman of the board of Public Library of Science and chair of the scientific board of the Bill and Melinda Gates Foundation Grand Challenges in Global Health. He also helped to guide the Cancer Genome Atlas that has identified genomic changes in more than 20 different types of human cancer. With this, I once again welcome you, and I request Professor Varmus to deliver his lecture.
Want to take questions? Sure. If there's anybody left in the audience.
Okay, so appreciate the kind invitation and Dr. Preetha's arrangements. The chance to be back here at the NII for the third time. Many years ago, I spent some, what do you say? What was that? Oh, yeah, thank you very much. And as you've heard, I spent some time in India, not far from here in Bareilly, UP, as a medical student, so I'm very fond of traveling back here, and pleased to be here. Even though I just got off the plane yesterday, I'm still feeling invigorated by the chance to be back in India. Today, what I would like to do is spend a little bit of time telling you about changes that I'm seeing in cancer research. And one way I find it useful to try to summarize these changes is to think about whether cancer is a simple problem or a complex problem, and whether it's one disease or many diseases. And there are many ways to think about that difficulty. I don't think we need to have the lights quite so low. People are going to fall asleep. Maybe we can raise the lights a little bit. Nothing I show will be that complicated. A long time ago, just about when I started doing cancer research, I entered the cancer research field in 1970. The following year, President Nixon signed the National Cancer Act. At a time of optimism about science, molecular biology was just coming into fruition. It wasn't yet the kind of powerful tool that exists today. We couldn't sequence DNA, we couldn't clone DNA. We didn't know a lot about the specific genes, but we knew there were tools for looking at how cells behave that were very powerful. And he was convinced by cancer advocates that with these new tools, we could actually conquer this disease. Importantly for today's discussion, many people gathered together and made a case for supporting not just individual cancers like breast cancer or cervical cancer, but research on all of cancers. And the idea of bringing those different problems together under the rubric of cancer as one disease was very useful, and it forms a background for thinking about whether we should think about cancer as a simple problem or a complex problem. If we think about it as a disease of cells, which it is, you can think that we know cells have programs they normally follow out. They divide, they differentiate, they undergo cell death, and that when cells become cancer cells, that is, grow in an inappropriate fashion, grow in inappropriate places, spread to different sites in the body, and end up killing the host by metastasis, we can think about those phenomena as aberrations of these normal processes, that is, growing too much, dividing inappropriately, failing to differentiate, failing to die, all of which will produce too many cells. And from that point of view, cancer looks like a pretty simple problem. That is, we have all of our organs are replenished from stem cells. All those cells go through these phenomena that are shown here on the slide. And if we think about trying to understand why cancers arise, we can think about it in these fairly simple terms. Of course, thinking about the physiology of cancer is different from trying to understand it at its fundamental level. And for me, coming into the field at around 1970, the big unanswered question is, how does a normal cell, the kind pictured here, become a cancer cell? What has changed to allow that cell to behave stably in a very inappropriate fashion, growing too much, invading locally, and spreading to other organs? And we had some clues. And those clues were very diverse. We knew that smoke, that cancer was associated with use of tobacco, with exposure to radiation, with exposure to certain chemicals. That can also be proven in animal models, particularly mouse models. And the chemicals that were used to induce cancer in a mouse, for example, were usually molecules that could be shown in other kinds of tests to be mutagenic, that is, they changed the sequence of DNA. We also knew there were some cancers that occurred in a familial fashion. They could be hereditary diseases. Sometimes cancers arose after infection of animals with viruses, and the assumption was that viruses introduce genes. Some cancers could be seen under the light microscope to have abnormalities of their chromosomes, and the changes that seemed to occur to make a normal cell into a cancer cell were stable the way mutations might be. So for all these reasons, even though there was a complex set of clues to how a cell might become a cancer cell, you might say there is a common potential theme, that is damage to the genetic information that's present in every cell in the body. But that gave rise to a deeper question, again, a question that might have a pretty simple answer, that is, if cancer arises from changes in genes, what are the genes that are responsible for causing cancer once they undergo mutation? Now, this was a difficult question because there were not very many ways to try to discover what's wrong. Today, you ask an average graduate student how to answer that question, and he or she would say, well, just clone the genome and sequence the whole genome and see if there are any changes. But in 1970, until only a few years ago, when sequencing techniques became very rapid and inexpensive, only recently was it possible to try to find the answer in a very simple way. And we'll come to that in a moment. But 45 years ago, there were very few routes that would allow you to discover what genes might be involved in causing cancer. And one of those routes, the one that I personally followed with my colleagues, was to make use of viruses that were known to exist in many animal species, viruses that contain their genes as RNA in the case of retroviruses, or in the form of DNA in the case of papillomaviruses and adenoviruses and a few herpes viruses. And those seem like potentially useful tools. Moreover, having the viruses themselves, which might not have been sufficient, was supplemented by other kinds of instruments that could be used for trying to ferret out, to uncover the genes that are involved, because there were ways to look not just in an animal, but actually in cells growing in a petri dish for that transition from normal cell behavior to cancerous behavior. Now, there were several viruses, as I mentioned, that could be used for such studies, but the one that I and many others chose was a virus that had been isolated in 1910 at Rockefeller Institute by Peyton Rouse, a young physician who was given a chicken with a big sarcoma, and he made an extract of that tumor and showed that it contained a particle which was already being called a virus. It passed through a filter. It was not a bacterium, too small for that, and could transmit a cancer to, or the causation of cancer to a new animal. And over the intervening years from 1910 to 1970, when my colleagues and I started working on this virus, there had been the development of simple assays in cell culture, like the one shown here, where Howard Temin, along with his colleague Renato Del Becco, showed that you could take basically a single virus particle and introduce it into an appearing normal cell of this kind that are shown in the background, and make what was called a focus of transformed cells. That was a powerful step in the direction of trying to understand how cancer arises, because now you have a simple virus carrying only a few genes entering a cell and causing that cell to display the kinds of behavior you see in cancer cells. The cells are abnormal in appearance, they overgrow the monolayer, they can grow and form a cancer in an animal. So this reduces the process of cancer formation to something quite simple. But it still raises the question of what kinds of genes are in the virus and whether those genes have anything to do with human cancer. And those are two issues I'm going to address in the next couple of minutes. Now, first, you have to have a genetic map. Genetic maps were quite simple in the 1970s. Here is a provisional map of the Rouse sarcoma virus genome around the mid-1970s, and there were at least 3 or more genes on the left, so-called 5' side of the genome, that were required for replication of the virus to make its coat proteins and its envelope proteins and its polymerase, the so-called reverse transcriptase, the integrase, the protease. But over on the right, there was a gene called SARC that had been defined by mutations, that either deletion mutations or point mutations, that affected the ability of this virus to cause the transformation in cell culture that I showed you a moment ago. So if the gene was completely deleted, the virus would grow because the replication genes were still intact, but the transformation gene was gone, and the virus could not cause the cell to transform. Even more interesting, mutants were isolated by using chemical mutagens to create so-called conditional temper-sensitive mutants of Rouse sarcoma virus. The most famous was one isolated by a postdoc in Berkeley named Steve Martin, who made a temperature-sensitive mutant of Rouse sarcoma virus that could be used to infect, a normal chicken cell, and when grown at a low temperature, the cell would transform and make virus particles, but at high temperatures, the infection led only to the production of virus particles, but not to the transformation of cells. What was extremely important about these experiments is that you could take the cell at high temperature and lower the temperature, and it would transform, and conversely, raise the temperature of a transformed cell and cause it to revert to a normal behavior pattern. And that suggested that the replication functions and the transforming functions of this virus were separate, as separate genes required for the two functions, and that the transformation was dependent on probably a single protein that would transform cells at a low temperature, not the high temperature, and that you needed to maintain the function of that protein by keeping the cells at a low temperature to maintain the cancerous phenotype. So all those results were very important in thinking about the kinds of genes that could make a normal cell behave like a cancer cell. And for many of us, the next question is, well, where does this gene come from? The virus really doesn't need it. The virus grows well at either temperatures. So our group set out to try to find out whether this gene might have been acquired from a normal cellular gene. And without taking you through all the experimental procedures, the bottom line was that yes, the SARC gene, the viral SARC gene of Rouse sarcoma virus, was a set of nucleotide sequences, a viral gene that was derived from a normal cellular gene, a gene that had been highly conserved in evolution, came like other cellular genes with introns and exons. The precise way in which it was captured, we understand partially. A very important feature of this is that in the process of capturing the cellular gene, the nucleotide sequence was changed slightly, and that was important because we now know that the normal cellular gene encodes a protein that has protein kinase activity that adds phosphates to tyrosine residues. That enzyme is very weakly active in normal cells. It is regulated by a mechanism involving a phosphorylation site near the C-terminus of the protein. But during the capture of the cellular precursor, the proto-oncogene, part of the gene was changed so that the regulatory elements were mutated, and now the gene present in the virus is a very active protein tyrosine activity that cannot be regulated. So that helps to explain why it serves as an oncogene. And that gave rise to the question of whether this gene, the cellular SARC gene, might be a target for the mutations that cause human cancer. Here we have a very simple problem in the mid-1970s. We identified a cellular gene that gives rise to a viral oncogene that makes cancers. Could the answer to this whole cancer problem just be a result of mutations that occur and affect a cellular gene called CSARC, and thereby explaining all cancers? But before we could try to answer that question directly, it became clear that the large number of retroviruses that exist created a level of complexity a lot higher than the simple statement I just made. There are many retroviruses found in animals that cause cancer in animals, and when my colleagues, and to a certain extent I and my close colleague Mike Bishop in San Francisco began to look at these other retroviruses, it was clear that there were a lot of other genes that, like SARC, had been captured from cells and had precursors in the cells that we call proto-oncogenes.